Perfect. I'll get started then. Hello, my name is Emma Fehlker Campbell, and today I'm going to be talking a little bit about how health biomarkers change in the presence of parasites, looking at the population of cottonmouths. So parasites are a common feature of any landscape. But with increasing human-caused changes to the landscape, we can expect changes both in the parasite community as well as parasite prevalence. And this can impact the survival of snakes, many of which that are declining across their range. So we're going to be looking at two main kinds of parasites. The first, as you can see here on the right, are trematode parasites. They're kind of a parasite that infects the digestive tract. And then secondly, we will be looking at hemoparasites, which are blood parasites that infect the red blood cells. We'll be examining two main health biomarkers. The first being concentrations of corticosterone in the blood, and corticosterone is basically just a hormone whose primary role is in the metabolizing of glucose, but it's also released as part of the stress response, so it's been commonly used as a biomarker of stress. Secondly, we'll be looking at the ratios of heterophils to lymphocytes, which are two different kinds of white blood cells found in reptiles. And the ratios between the two change both with stress, and they kind of serve as a marker of immune function. So we have done this research on three main sites here in Western Kentucky. The first one being Clark's River National Wildlife Refuge, which we sampled in the spring of 2020. The second being Murphy's Pond, and the third being Terrapin Creek, which we both mainly sampled in spring of 2021. With each snake, we would draw a small amount of blood, like 35 milliliters, within 5 minutes of first detecting the snake to get a baseline concentration of the corticosterone. We'd also use the blood to make a thin blood smear, and with the blood smear, we would stain it and use it to count the number of hemoparasites per every 1,000 red blood cell, and then to get the heterophil to lymphocyte ratios, and that's a heterophil with the H pointing to it. So here are some of our preliminary results. So first of all, we did not find any strong relationship between either type of parasite and the corticosterone concentrations. But here are some things that we did find. So first of all, we saw a pretty large difference between the percent of cells that were infected with hemoparasites between males and females. And we think this is largely because of increased contact between the males as they're fighting for mates. And then most of the differences that we saw were largely site-based. So there was a higher concentrate level of hemoparasite infection at the Clark's River site, and then there were higher levels of trematodes as well as higher corticosterone concentrations at the Terrapin Creek site. And we think that this is largely based off different characteristics of the sites. The Clark's River site is larger, it's less fragmented, and it has a higher population than the Terrapin one. And that higher population is just increased contact between snakes, which means more hemoparasites. And we think that the higher corticosterone concentrations and the trematodes are just because the Terrapin site is smaller and a lot more fragmented. It is important to note that with the Clark's River site and the Terrapin site, most of the snakes that we did, that we sampled across different years. So it's hard to tell if this is exactly site related or year related. But we did end up sampling two individuals from Clark's that second year, and they had similar levels of parasitism and corticosterone concentrations to the first year. So we think it is largely based on site instead of year. So overall, we have not found a really strong relationship between cord concentration and parasite loads for either one of our parasite types. And most of the variation that we saw was based on site, most likely. We currently are working on finishing the heterophil to lymphocyte ratio counts, and that is something that we are wanting to include in the future once we get that finished. I'd like to thank everyone for their support for this project, for everyone who helped me in the field, and I'd like to thank the Superb Scholarship Program, the Watershed Studies Institute, and Murray State University for their funding. Thank you.